Today we're going to talk about the transport of the respiratory gases, oxygen and carbon dioxide, around the body. Let's look at oxygen first. Although oxygen could be transported by plasma, there are a few reasons why it's not the best method. Plasma has a low solubility, and its solubility is affected by temperature increases. It's sensitive to these. Therefore, it's an inefficient way to transport enough oxygen around the body. Instead, oxygen is carried by haemoglobin, a molecule contained by erythrocytes or red blood cells. Their structure supports the function of efficient transport of oxygen around the cells of the body. The structure of haemoglobin is that of a quaternary structure. Therefore, it's made up of 4 polypeptide chains. So you can see here we've got number 1, number 2, number 3, and number 4. And these are 2 alpha and 2 beta. And these 4 polypeptide chains each have a heme group. And you can potentially see them with these 4 little yellow discs. Each heme group contains an iron atom that can each bind to a molecule of oxygen. Why can haemoglobin not run wild? Why does it have to be contained within a red blood cell? Well, for one, if it were free, it would mean that blood would become too thick and it would impede blood flow. But also, being enclosed within a red blood cell prevents the haemoglobin from being filtered out of the blood by the kidneys and lost. It's also important to mention why and how oxygen gets picked up by haemoglobin. Concentration gradient and the principles of Fick's law play an important role in this, but we'll talk more about that later. We've already discussed that the structure of haemoglobin supports its function of carrying oxygen. In theory, because of the presence of 4 heme groups that can each bind to an oxygen molecule, haemoglobin can carry 4 oxygen molecules. If a haemoglobin was to carry its maximum capacity of 4 molecules of oxygen, we would say it's completely saturated. Haemoglobin that's carrying oxygen is referred to as oxyhemoglobin. However, often haemoglobin doesn't carry 4 molecules of oxygen. To explain why, let's look at an oxygen dissociation curve. The x-axis represents the partial pressure of oxygen. You can think of this as how much oxygen is around or its concentration. And the y-axis represents the saturation of haemoglobin with oxygen. It appears as a sigmoid curve. And the curve represents how oxygen binds to haemoglobin, and we can see that this occurs unevenly when exposed to different partial pressures of oxygen. At the bottom left-hand corner of the graph, we can see that there is a low partial pressure of oxygen. This, along with the original shape of haemoglobin, makes it difficult for the first molecule of oxygen to associate. However, once the first molecule is associated, the conformation of haemoglobin changes. This makes it easier for molecules 2 and 3 to associate. This is often referred to as positive cooperativity. And this can be seen on the graph here. So molecule 1 starts to bind, and then the exponential rise is molecules 2 and 3 binding. However, after 3 molecules have associated, the haemoglobin structure has become distorted so much that it's difficult for the 4th oxygen molecule to associate. And this is why we see a plateau on the graph. Now, I said we'd come back to the importance of concentration gradient, so let's get stuck in. In order for cellular respiration to occur, we need to get oxygen from the air to the tissues. This is enabled by a respiratory and circulatory system working together, facilitating mass transport and simple diffusion. In assignment 1, we looked at mass transport, and we know from previous studies that for simple diffusion to occur, the principles of Fick's law must be applied. So, we need a short diffusion pathway. The structure of both capillary beds and the alveoli at the lungs are 1 cell thick. and then flexible red blood cells squeezing through little gaps and pressing up against the capillary walls shorten this diffusion pathway. We also need a steep concentration gradient. Remember that molecules diffuse from a high concentration to a low concentration. Therefore, we need a big difference in the partial pressure of oxygen where it is being exchanged. And we need a high surface area to volume ratio. Again, the structure of both alveoli in the lungs and the red blood cells themselves all contribute to this. So as blood returns to the heart and then to the lungs, the partial pressure of oxygen will be low, and this is due to the hungry respiring tissues having used it up. This makes it easier for oxygen molecules to diffuse from the alveoli, where the partial pressure of oxygen is higher due to ventilation, into the blood to be picked up by the haemoglobin. And we can see this here. So we can see that alveoli are covered in capillaries. Of course, we've talked about structure and function of blood vessels before, and how with the principles of Fick's law, they support diffusion. As inhaled air comes into the sac of the alveoli, we have a high pressure or high partial pressure of oxygen, and the deoxygenated blood that's arriving in the capillaries and within the red blood cell, there is a low partial pressure of oxygen. So therefore, with this short diffusion pathway, only 3 cells thick, and with these steep concentration gradients of oxygen, we are able for this diffusion to take place. Now let's think about when oxygenated blood arrives at the site of hungry respiring tissues. The partial pressure of oxygen will be high within the red blood cells, as we know from earlier that it's now full of oxyhemoglobin. The partial pressure of oxygen within the tissues will be low because the oxygen has been used up via cellular respiration. This creates our steep concentration gradient, and ta-da, diffusion can occur. However, this is not the only factor that affects hemoglobin's affinity for oxygen. 2 byproducts from respiring cells are heat and carbon dioxide. The Bohr effect explains how the presence of a high concentration of carbon dioxide negatively affects hemoglobin's affinity for oxygen, and how hemoglobin's behaviour changes at different areas of the body so that respiratory gases are efficiently loaded and unloaded. To understand the Bohr effect further, we first need to know 2 things. We need to remember that hemoglobin is a protein with a quaternary structure. Therefore, we know, because it's a protein, that fluctuations of pH and temperature cause conformation changes, and this in turn will affect hemoglobin's affinity for oxygen. Secondly, we also need to know what happens to carbon dioxide. There are 3 ways carbon dioxide can be transported around the body. So one way carbon dioxide is transported around the body is that a small amount of carbon dioxide is dissolved in the plasma. This is only about 5%. Secondly, some carbon dioxide diffuses into the red blood cells and binds to hemoglobin molecules to form carbaminohemoglobin. However, lastly, approximately 85% of carbon dioxide is transported as hydrogen carbonate ions, which are formed within red blood cells but transported within the plasma. Let's have a quick look at this third process and how that occurs. Carbon dioxide produced by respiring tissues diffuses into the plasma and then into the red blood cell. This carbon dioxide reacts with water, H2O, and produces carbonic acid. This reaction is catalyzed by the enzyme carbonic anhydrase, and it is a reversible reaction, hence the arrows either way. So carbonic acid is H2CO3. The carbonic acid then dissociates to form hydrogen ions, so H+, and hydrogen carbonate ions, so HCO3-. Since the hydrogen ion is acidic, to prevent a drop in pH, hemoglobin binds with it to act as a buffer, forming hemoglobinic acid, so HHB, hemoglobinic acid. In order to efficiently transport carbon dioxide, it's also important to process the hydrogen carbonate ions. Therefore, it gets transported outside of the cell and into the plasma. However, this creates a shortage of negatively charged ions inside of the red blood cell, which needs to be balanced. Therefore, chloride ions, so Cl-, move into the red blood cell, and this is called the chloride shift. The reaction of converting carbon dioxide quickly to carbonic acid and then to its subsequent hydrogen and hydrogen carbonate ions prevents a buildup of carbon dioxide in the red blood cell and maintains the steep concentration gradient so that carbon dioxide can continue to diffuse out of the cells. Got all of that? Good. So back to the Bohr effect. In a high concentration of carbon dioxide, the hydrogen ions that are a result of the carbonic acid dissociating slightly decrease the pH. This creates a conformation change which displaces the oxygen from the oxyhemoglobin. The oxygen moves down its concentration gradient to the respiring tissues, and the leftover hemoglobin mops up the hydrogen ions, forming hemoglobinic acid and acting as a buffer to maintain a fairly neutral pH. This is known as the Bohr effect, and it's the mechanism within the body that explains how respiratory gases are efficiently loaded and unloaded. The Bohr effect causes a shift to the right on the oxygen dissociation curve known as the Bohr shift. So that wraps up our little video on how respiratory gases are transported around the body. We have looked at why oxygen is carried by hemoglobin within the red blood cells and how it does so, the importance of partial pressure gradients, paying special attention to Fick's law, the oxygen dissociation curve and its shape, the Bohr effect, the Bohr shift and why it occurs, how carbon dioxide is transported around the body, its three ways and why it's important to prevent a buildup of carbon dioxide within the red blood cells. My name's Emma. Thank you so much for watching.